So I had total who took the test, 50 total. And that again, how
I
many?
had 26 students take the pre-assessment.
And this is what we're going.
I had 17.
Proficient or higher on the assessment, and for me was 27. My kids
I had 14 that were not proficient, so that would be 12 that were proficient.
I had 9 that were proficient.
non-proficient. Abraham is 23. That again.
14 for Latigan.
Oh, that were non-proficient?
Six.
Total minus 9.
72 minus 9. 63?
So we should have someplace here where we can just write it as a percent proficient. I can just write it here.
That's fine.
Did I need a percent?
Well,
Just
here.
for us, because
Yeah.
we're going to compare it.
Oh, sure.
Proficient is
Huh. I had 13% proficient.
Mine is like calculator.
How many?
26. 46% was Latigan.
I had 13%.
Alright, strength and obstacles.
Alright, so we got these data entered. We got some students that are not proficient and we need to get them proficient. So let's discuss some strategies that we can use. I know when I analyzed my data, we assessed them on these functions of organelles, which is basically definitions, and then we had a diagram for them to identify those parts within the cell. And my students overwhelmingly struggled on the functions or the definitions of the organelles, and they were better off on the diagram section. So the visual stuff they seem to be getting more so than the definition stuff. So I did an activity personally today that had them playing a game where they were matching definitions to organelles, like that memory concentration game, and they kind of had to work with it in pairs, and they were competing against each other to see who could get the most matches. And I had a sheet that went along with that reinforced the functions where they had to rewrite them again. So they were seeing them in the visual context of the game, and they were writing them on the sheet separately. I think the cell project we did in my class really helped them with the visual stuff. They seem to be able to label the diagram much better as a result of having to build the cell themselves. When I talked to each student about why they didn't do so well in the definition part, and a lot of them just admitted, Mr. Latigan, I didn't study the flashcards that you assigned, or I didn't do the flashcards you assigned, or I didn't complete the homework that you assigned. They requested more sort of interactive with their peers type of activities to help them learn it. That's why I did the game today in class. And tomorrow I'm going to be doing all group work tomorrow, more stuff with like analogies of making an analogy to the factory and things like that to try and get those functions to stick into their head. think analogies can be really useful for that. Um,
And that goes to our obstacles, because number 2 is analyze strengths and obstacles. So I think one of the obstacles that we've all seen is that they're just not studying outside of school. And this is such a language-rich concept and subject altogether that they need to spend some time studying, and we're not seeing them do that. And admittedly, they're not
Yeah.
doing that. Yeah. So that's definitely an obstacle that we need to then try to address in the classroom, that we can't count on them for studying outside the classroom, which is what we were talking about.
Yeah.
So if I felt like the memory game we played today in class was useful for them it's kind of studying in sort of a game sense. So if you guys would like, I have that pre-made up in my room if you want to
Great.
borrow it for
I do
one
the
day.
notes too with they take the grid, it's a notes grid, and then they have to pick which picture goes with that organelle, cut it out. I give them pictures already. Cut it out and put it there. My regular class, I give them the structures and the functions, and they have to cut them out and put the structure down and the function down, and then they have to define it using those two. So that's how I give them notes on it.
I would say that under here that those are both advanced organizers, cooperative groups.
I assigned mine flashcards to make flashcards at the beginning when we started talking about this, and as an anchor activity for anyone who finishes work early, I say go study your flashcards, because this is it's a lot of study. You really have to see the material repeatedly to learn all the different functions. It's not so much a difficult concept as it is just vocabulary, like you said. What's another strategy we could use to bump up vocabulary? I heard one of you mention Latin stems earlier.
Yeah, I use Latin stems on a daily basis. And when we do things like cytoplasm, they know that cyto means cell and plasma is liquid, so cytoplasm is the cell liquid. No need for a long definition, you can break it down. So you can break down the vocabulary with the Latin stems, you can look at the pictures and break it down and get to the structures and the functions from that as well. So there's no reason why they shouldn't be getting it. They just have to do it. But I give mine Latin stems every day, and then we just go back to them and then reference the ones that pertain to that chapter.
So Latin stem seems to
I'm
work
gonna put that
somewhat.
under, I think that's creating, the way that you do it. Creating, comparing, classifying, creating, summarizing, effort, investigate, prove, hypothesize, writing, inferring, making
I
observations.
think it's inferring. Maybe inferring and
Yeah, they
creating.
kind of infer the definition based on the root of the word. As far as I guess, is there one that's like synthesizing or?
That's creating.
Creating.
Oh, creating, yeah.
Yeah.
As far as creating goes, like I tried this thing where they each had to make and perform a commercial that advertised their cell as a product that someone would want to buy, and what are the benefits of that cell organelle and why you would need it if you didn't have it. And I feel like some of them came up with some creative ideas for sticking those into their head for memory. And I try to reference those commercials in class when I say the Golgi apparatus came up. I say, do you remember the commercial that these two students did when they performed the Golgi apparatus? And a lot of them were like, oh yeah, I remember that. And so having them sort of create their own definitions or functions in a sort of outside of classroom context can be useful as well.
And did you do cell coloring?
Yeah, I had them color in the cell diagrams, and I think that's
comparing.
partly why they did better on the sort of diagram section, because they really had that colorful visual to to
Which
think
was-
back to.
Prokaryote and then plant and animal, bacteria and then plant and animals.
Mhm.
To
Yep.
compare the three.
So we've talked a lot about
Yeah.
stuff that we have done that seemed to work. Is there anything that we can still do in the future to get these kids more on the proficient side of things?
Just looking at instructional strategy, some way of summarizing or writing where they have to pull all the ideas together. They do seem to miss that bigger idea, which is that there are two main types of cells and here's the difference between them, and then of the more advanced cells there are two types and here's the difference between them. Like they can do it as rote memorization if they've memorized it, but I find that they still don't see that bigger picture. They couldn't necessarily summarize it the way we could.
Have you tried that graphic organizer that you
I
had
just,
up there?
I got that today, and only my kids who finished other work did it. So, they haven't, yeah, it starts off. And the only two kids looked at it, and one of them was like, he'd never seen this picture before, even though we've been spending it. So it does challenge him in a different way.
Oh,
Just trying to sort that information.
yeah.
Yeah, so this graphic organizer kind of takes those little pieces and puts them together in a bigger idea for them to sort of
Yeah.
map it out in their heads.
The POGL number 9 for the eukaryotic cells also said trace from the center of the factory. If the factory leader gives directions for the product to be made in the factory, trace that through the factory. In other words, trace it from the DNA out of the cell. Your product that you're making. So that's a really good synthesis
Yep.
idea too.
Are you doing that? Did you do that?
Yeah, we're in the process of doing it.
I'm gonna put that in summarizing, because I think.
And they get
that's
stuck
a
on
good
it because they
new
can't
one.
go and make a story with all the organelles.
Okay, so to summarize, basically we've identified that they are struggling to learn the functions of the organelles, partly because they are having trouble connecting the small pieces to the big ideas. So we're going to try a graphic organizer to sort of connect those big ideas together. We're going to try making analogies for all the functions to connect them to real world ideas that might already be in their framework, so it's not all memorization from there. And a lot of group collaboration where they can sort of help each other, because I know some of them have weird ways of remembering things that they can share with their peers that may be something we wouldn't think of as the teacher. And we also just want to encourage them to study outside of class every night a little bit and provide them with the tools they need to study, such as flashcards and the Latin stems that we talked
The
about.
Quizlet, which is essentially a cyber flashcard.
Yeah, we provide them with the online study tool like Quizlet.
We also did make observations. I know we all looked at least at plant and animal cells under the scope.
Yeah.
Looking at the microscope. I don't know how much they took away from that, but it was something to
We're doing the onion cell, cheek cell tomorrow. Yeah. So they learn how to make the
they're gonna make the
slides too.
Yeah. Cool.
Yeah.
So maybe seeing the cells under the microscope will help them connect the definitions to their real world application.